Section 10.3 is about how the cell cycle is regulated. We've learned about what the cell cycle is. Now, how do cells figure out when to divide? The controls on cell growth and cell division can be turned on and off. When there's a broken bone or a separation or a cut between tissue or in the middle of a tissue, the cells then are stimulated to divide and start healing. Once cells start to come into contact with one another and the healing process is near completion, cell division slows down. Proteins called cyclins are the ones that regulate the timing of the cell cycle. The graph here on this slide shows how cyclin levels change in fertilized clam eggs. So the cyclin concentration is on the y-axis minutes after fertilization here. So you see the peaks of cyclin concentration during mitosis when the cell is dividing. There are different ways that the cell cycle can be regulated. Internal regulators are proteins that respond to events inside a cell. So they have these checkpoints that they monitor. The cell cycle can proceed only once certain processes have happened. Has the DNA copied itself? Do we have enough of each chromosome? Is anything missing? External regulators respond to events outside the cell. And they tell cells whether to speed up or slow down the cell cycle. Growth factors are one example of external regulators that stimulate the growth and division of cells. They're important during embryo development and wound healing. If cells aren't working properly, if they don't have the proper content, if they're not working right, if there's a problem with them of any kind, they're basically supposed to check themselves out. Apoptosis is the fancy word for that, programmed cell death. What happens with apoptosis when in embryonic development, tissue development, the hand with digits, or this says the foot of a mouse, and there's my artistic ability again. At one point, there were webs between all these fingers, and these cells in between, right here, right here, right here, right here, died off. Apoptosis, programmed cell death. So it helps shape structures, like between the digits, the way it's supposed to. Gets rid of the webbing. And also, like I said, if cells aren't functioning properly, they should also check out as well. Cancer is when cells lose the ability to control the cell cycle. It's basically uncontrolled cell growth. The mass of cells that forms is called a tumor. In normal conditions, cells that have lost this ability should undergo apoptosis. But this goes back to the M word, mutation. Mutations accumulate in the cells, and if that mutation occurs within genes that are supposed to control the cell cycle, then the cell cycle is not controlled and you have uncontrolled cell growth. Mutations cause cancer. It causes the cells, our own cells, to become different from what they're supposed to be. They grow out of control. They don't have the ability to check out on their own, do apoptosis. This is when you see problems arise. A benign tumor is non-cancerous. It does not spread to surrounding healthy tissue, just basically an extra ball of cells. Malignant means cancerous. It can invade and destroy surrounding healthy tissue. It can spread to other parts of the body, which is called metastasis. And cancer cells are basically parasitic. They absorb nutrients needed by other cells. They can block nerve connections and prevent organ function. So really bad news. Like I said before, cancers are caused by defects, by mutations in genes that regulate cell growth and division. Gene defects or mutations can be caused by what we call carcinogens. Carcinogens are things that can cause cancer. I spoke very generally about mutagens the other day, which cause mutations. Carcinogens are specifically mutagens that can cause cancer. Some things which you probably already know that can cause cancer: smoking tobacco, radiation exposure. People can be born with mutations already, defective genes, mutations in their genes. And viral infections can also do that. A common mutation is in the gene called P53. P53 plays a role in apoptosis, so if it is not working properly, it can lose the information needed to respond to growth signals. Cancer can be treated by surgery. They can be targeted with radiation or chemotherapy, which is medicine that's injected into the bloodstream. The rationale here with the radiation and with the chemotherapy, both of these are going to kill healthy cells as well. But chances are, what we're banking on is the chances that there are fewer cancer cells than healthy cells. So yeah, we're going to kill some healthy cells, but we're going to kill all the cancer cells. That's what the hope is.